Thank you everyone for joining us for our annual video series from the International Symposium on Human Identification. We have a special guest with us today. Ellen, why don't you tell everybody a little bit about yourself and your work?
Hi, I'm Ellen Greytak. I'm the Director of Bioinformatics at Parabon Nanolabs. These days, I think we're mostly known for doing genetic genealogy, which is awesome, but we do a lot of stuff other than that. We started out with DNA phenotyping, and we also work in medical areas, in non-forensics. So we do a lot of different projects, but the general theme of our work is, given a DNA sample, what can we say about that organism?
Wonderful, thank you. Well, why don't we talk a little bit about the poster you submitted for this year's symposium? What was that?
The poster that I did this year is called DNA Phenotyping Using Ancient DNA from Ancient Egyptian Mummies. So this idea came about. We found that there's a lot of data freely available online from the ancient sequencing projects that have been going on. And it was sort of a question of, could we get the data to an appropriate level that we could actually do DNA phenotyping on it, on someone from thousands of years ago. So it's sort of a proof of concept, but these same tools that are being used for the ancient DNA can also be applied in forensics, where we have very difficult samples as well.
That's fascinating. How do you use the techniques that we have for DNA to gather the genomes of people that lived thousands of years ago?
Yeah, the whole field of archaeogenomics is really fascinating, and it has come a long way since the Neanderthal genome was first published. Now there are thousands and thousands of ancient sequences out there that have been published. And so there have been innovations both on the laboratory side and on the bioinformatics side. On the laboratory side, there have been developments in, for example, capture assays, where a big problem with ancient DNA is that when you extract that sample, most of the DNA you find is actually modern bacteria, and that DNA is in much better shape than the ancient human whose DNA you're trying to analyze. And so these capture assays have baits that actually pull out the sequences that belong to human, and in that way you can sort of enrich for the sequences you're interested in. You still are going to be sequencing some bacteria, but you can actually pull that human proportion up from where it may be, below 1% up to several percent, or even a few tens of percents. And in addition, on the bioinformatics side, being able to go from these low coverage genomes to really high quality data, that's something that's just come about in the last year or two. I'm sure you've heard Ed Green talk about it. This is low coverage imputation, the idea that we can go from really low coverage genomes, and the DNA that we had in this case, they were all far less than 1x coverage. And yet we were able to apply low coverage imputation to actually recover most of the SNPs that we needed for DNA phenotyping. So we could basically take something that's covered at less than 1x and get hundreds of thousands of pretty accurate SNPs from that. And so, like I said, this is sort of a proof of concept in this cool idea of ancient Egyptian mummies, but those same techniques are also being used in our forensic cases.
That's fascinating. So now that you're able to gather this high quality data, what does that mean for people who are studying ancient cultures, or let's maybe also what that means for those using forensic DNA for other purposes?
Well, I think ancient DNA has really changed the face of anthropology, because for a long time, the only way we could learn about how cultures interacted was trying to infer from what we had found. And now we can actually directly see how the DNA flowed from one place to another in different time periods. It's been really fascinating to see. I mean, I feel like every day there's a new paper coming out with new ancient genomes, comparing them to the modern populations that are there and able to tell the story, the demographic story of the human population.
So what are some of the challenges you faced as you're doing this? I mean, every year we're taking a leap forward, but not without a lot of hard work. What have you learned along the way?
Well, these data sets are really, really tough. And definitely a few years ago, we were of the same opinion as everyone else that if you had less than 1x coverage, there really wasn't a lot you could do. You could call a few SNPs here and there where you got lucky, but you really couldn't put together a high quality genome-wide covered set of SNPs, but now we know that it is possible thanks to research that's been done and is out there. And so it's just a matter of sort of implementing that pipeline. But we are still seeing some data sets there's only so much you can do. So with the poster that I'm showing, there are 3 samples. One of them had close to 1x coverage, and that we were able to get beautiful data out of. The others were much lower. And so we actually were not able to impute nearly as many SNPs. And so we had to do a little more guesswork on the phenotypes, which means we have much bigger confidence intervals. And so it's sort of a good use case of, you know, what can and can't we do with these types of data.
Well, talking about the phenotypes, what were you able to, in this particular case, what predictions were you able to make? What did you find?
It was pretty neat. In the original paper on where they published these sequences, they had looked for a known SNP for fair skin color in the higher coverage sample, and they had found that it was there. So they thought they probably had lighter skin, but that was the extent of it. So we did find that as well. Unfortunately, like I mentioned, we were not able to get all of the SNPs that we were interested in. And there's one particular SNP that's very, very important for eye color, and we were not able to get a genotype for it in the 2 lower coverage samples. So basically, I made predictions for them, assuming multiple different genotypes. And so they did all come out as fair-skinned and brown hair and dark eyes. Interestingly, the ancestry, I think the ancestry was the most interesting part. They really did not cluster with modern Egyptians, but instead with people more from the Arabian Peninsula, more up into the Middle East rather than Africa. And so that was also what had been found in the paper, that there really was no sign of sub-Saharan African ancestry in these individuals. And so that must have come in during a later phase of the Egyptian population.
It's fascinating that you can confirm some of these theories or add new evidence to the case. That's really amazing.
Yeah, it's very cool. And so we're now looking for, you know, what other genomes are out there, and thinking, I mean, genetic genealogy, well, for ancient samples, we wouldn't really expect to be able to find anything. But if these same techniques, we've been using them in our casework, and we are able to do genetic genealogy on really difficult bone samples where there's a lot of microbial contamination, not a lot of coverage of the genome, and we're getting beautiful, nice imputed data sets. And so part of this poster is also showing a validation of the imputation process and showing how we can go from really low coverage to really nice data.
It's really remarkable. How did you get involved in this work in the first place? I always like to ask that.
Well, I did my PhD in evolutionary biology. And after my postdoc, I was just looking for a job and happened to see a posting of, you know, we have this grant to predict what people look like from DNA. And then we're looking for a biologist to lead that project. And I was like, that sounds like just about the coolest thing I can imagine. So it was really very fortunate that Parabon was looking for someone when I was looking for a job, because I think it's so much fun. I mean, every day I get to learn something new. So I love my work.
Any recommendations for people who might be thinking about entering the field?
For me, the biggest change, and so obviously things have changed a bit in the last 10 years since I graduated, but learning how to program changed everything. When I got out of grad school, I could not find a job. Nobody really wanted an evolutionary biologist. But once I learned how to program, and it became not just I can answer some questions, but I can write something, I can create something new to ask new questions. All of a sudden, everything opened up. There were so many options out there. And so I always tell anybody, if you are looking for one skill to work on, it's learn to program.
That's a good tip. I don't know that we've had that tip before. I think that could help a lot of people. Can you talk about what you're working on now?
Well, we're working on lots of different things. Of course, we always have the casework going on. Right now, the big project that I'm working on is actually studying antibiotic resistance in bacteria. So analyzing the genome of a bacterial strain and predicting what antibiotics it would be resistant to. So that's what I'm up to these days.
Wow, that's fascinating, especially in light of everything we've just been going through.
Oh, yeah.
Absolutely. Yes. And the reason we're digital, as people can see, for a little bit longer. Anything else you want to talk about, like maybe what the future holds for phenotyping, given what you've seen, the progress over the last couple of years?
The literature has been full of really cool papers, both expanding the populations for phenotyping and expanding the types of phenotypes that we can do. I mean, I'm still really excited about the idea of being able to predict age from methylation. I feel like I've been saying that for about 5 years now, but I'm really hoping that one day it'll start actually being able to be used in forensic cases, because that could really make a huge difference, not just for phenotyping, but for genetic genealogy. I mean, knowing the generation of the person you're looking for, instead of it's got to be someone between 14 and 64 or something like that, that could make a huge difference for a lot of cold cases.
Absolutely. I will look forward to talking about that in the future. Ellen, thank you so much. We really appreciate you taking time, and hope to see you at ISHI in person in the not too distant future.
Me too. Thank you.
Thank you. Have a great day.
You too. Bye.
Bye-bye.